So thanks for the organizer. It's really great to be here. It's actually the first time I'm talking in Paris, which is like really bizarre for me. So it should come more often, obviously. So what I'm going to talk today is about how, personalized medicine, combined with innovation in clinical trial design and how these two things are playing out. With a strong focus, of course, on adaptive trial design because that was the focus of the session, but I will go a bit further than that to show you at least one example about how precision medicine impacts clinical development in general. I forgot my disclaimer slide, but I'm from Roche, so you can imagine. Right, so quickly, introduction. So, when we talk about innovation in drug development or clinical trial design, there are three major areas that are really on top of our mind right now. The one on the right, which is obvious, is constantly thinking about making development as efficient as we can do. And so that's back to the same thing that Paula was saying before. Reduce in as much as we can the development time to get drugs or medicine to patients that are in need as fast as we can. The other one is, of course, patient-centric. There's a whole session that I'm looking forward to during this session or later on. It's really about meaningful endpoints, but also care about the patient during the trial and while they're on medicine, and I'm going to show you an example of that. And the one that has been constantly investigated but really becomes, to my great pleasure, much more prominent is patient diversity. We should aim at treating everyone. And so, of course, what it says behind that, you can see on the bottom that personalized medicine play a key role in every aspect of these three pillars. The obvious one, patient diversity, if we think about ethnic background, it's not so much about making sure that our trials are representative. It's about studying whether the disease progression is the same between ethnic groups and, tackling that when you're doing drug development. So it's clear that precision medicine feeds into these three pillars. If we go a bit more in detail, innovation in clinical trials, like this, you have to see that as a kind of impressive toolbox, the things that you can do and constantly adapt to actually the questions you have and your needs and constantly having in mind, again, the three pillars I was having. I'm going to quickly go through that. So the first one is, of course, how you conduct trial and patient centricity. You see that on the right. Many things there that you can do. The one thing that I will mention, because I won't have time to talk about today, is about screening patients for inclusion in clinical trial. There are very, very nice things that are, of course, existing in cancer, but I will cite Alzheimer there, where we see more and more development in terms of non-invasive screening of patients for potentially going from mycognition to AD, which is like the current target of population target for drugs. Which is helping patients not to go through MRI or PET scan in order to confirm that they will likely to progress. So that's what I mean by that. So having a more focused patient group to really have your efficacy readout. And I will mention the safety monitoring in that. Was said before, everything around innovation in clinical trial comes with new data type. We had an amazing example this morning. And on that, I just can't wait to have the results of the ML-based features on H&E slides at scale, because then we can have so many more information on the trial because we can have them, and that could enable to completely revisit everything we know about response to immunotherapy, for example. So that will not happen in the next 10 years, and we're talking about the next 18 months. But there's many more things outside of oncology. Of course, digital health technology. In neuroscience, again, will play a major role, but image analysis for ophthalmology and so on and so forth. So we're constantly fed by new data types that we need to understand, analyze properly in order to drive decision again. New data sources, we won't talk a lot about that because that's the talk of Krishna. The only thing I will mention there is that, of course, there are real-world data and so on. But we should forget efforts that we're doing in terms of system pharmacology, so understanding from cohorts, how the disease is behaving, abstract that, so that we don't have to have control arm anymore, but simply models that can help us looking at efficacy of the compound. The one on the left is the obvious. We can do more personalized trial. We see an amazing example from Paula, platform trial. There's master protocol, umbrella, basket, and so on, and adaptive trial design. And that all goes to, again, different disciplines coming together about the best way to analyze this data. That's very clear. I'm going to talk from a stat perspective on adaptive drug design, but there's many, many more things that are happening. in the background, as you can imagine. All right, so adaptive trial design, and in bracket enrichment, because that's why personalized medicine is about. Some concept, you've seen already a very nice example, and then some consideration not to forget about how to do it right, because they are rarely free lunch, as we know. Okay, so quickly, definition coming from like both the EMA and FDA, more or less you change something to your design as you go on the trial. That's more or less what it says. So, okay, that's why it's adaptive. There's no real magic in there. Okay, why we're doing that? So the first one is that in period of uncertainty, when you start your trial and you don't know exactly what's the right population or the right dose and so on, you can have a sequential approach, like testing different dose group or different subpopulation, or you can do it at once. And what it comes down to, if you do it right, is efficiency. So increased power, smaller trials, or shorter trials. And of course, it's more ethical because you have boundaries and decision rules that allow you to stop certain arms or certain trials if it doesn't show any benefit. And then it's more generalizable. Because you're testing different things and you test them right, your conclusions are more robust to a larger population, larger dose group, and so on. And it adds flexibility to the system. You can run basically different trials in one. Okay, so adaptive trial design didn't start by precision medicine. It's something that we are doing, I think the concept is from 98 or something like that, or 99 formalized, but has always been part of the drug development. The very basic one is dose finding. You start with a drug, you don't know what's the exact dose that's going to be efficacious, so you test different of them, you may stop arms as you go, and focus on the one that has the most promising result. And you can do many things on that. You can re-randomize between your control and your active treatment if you're not sure about what's the right power in your trial. But the one I'm going to talk about is, of course, the enrichment, which is core to precision medicine. So there are two setups. Paola has been beautifully exemplifying the phase 2, which I would still see in the early clinical development setting. It's all about signal seeking. What she was saying in her talk is like you're looking for big effect. Your primary goal is really to test the promising drugs that you want to go to phase 3 and beyond. So it's really signal seeking. Data can be blinded or not, so that can be a phase 1 and then without control and without randomization and so on. You can allow yourself to continuously look at the data if you want, so because it's not about type 1 error control. And of course, you can play with different things about, again, basket, umbrella, platform trials, as was described before. So, and you can do many things. You can do the enrichment, you can do the dose finding, you can change the endpoint. You can play a lot on that, and it's all going well with a Bayesian statistic, of course, the probability that your treatment is above a certain threshold. When you do that in confirmatory, so you have in mind that your trial is going to lead to filing, then it's really becoming serious. So you see like very serious terms in red, like integrity, validity, protection, review, and so on and so forth. So what it means by that is that you have to start, of course, by a clear hypothesis, and you need to make sure that at the end of the trial, the conclusions you're making are really back to what you're saying and that you can make these conclusions because they're going to go in patient at some point. So that's about the integrity, the randomized blindness, and the validity of your conclusion. It needs a fair amount of pre-planning because this trial can become complex. And the one and only thing you cannot compromise upon is the type 1 error. You need to make sure that if you make a conclusion, it has the right level. And the last thing, of course, you don't look at the data when you're the sponsor. Has to be reviewed by an independent data monitoring committee who will apply the decision rules that can be complex and doing that with them. And of course, those all have to be under confidential agreement because otherwise you can run into trouble as you go. So the example I will go is actually in TNBC. It's actually neoadjuvant, so we see the same thing, pathological complete response, and we didn't align on that, but it turns out it's the same. It's on the competitor of Pembro, who is not the standard of care, but that's the drug we have, so that's the thing I know. No, it's still on market, so I shouldn't say that. The story was the following. So TNBC, neoadjuvant setting, pathological response. We start originally by a simple, fixed, randomized clinical trial. And then as we go, and that was again what Paola was saying, more and more data were coming from PD-L1 expression saying that it could have either a prognostic or a predictive effect on atezo. And that wasn't clear at the point. But the only way not to have another trial in the PD-L1 positive population was to adapt the setup so that we could actually test for that. And so while the trial was ongoing, it became adaptive. So it's a simple case. And I purposely spent some time on the simple case to tell you that the simplest case is actually not so simple. So you start by stage 1. You have two subpopulations. F is the full, so it's the ITT neoadjuvant population, and S is the subpopulation, so the one that has PD-L1 positive in our case. You have two stages, as you see, stage 1, stage 2 at the end. And then stage 1, you can test 3 different hypotheses, whether like both F and S are significant, whether F only is significant, whether S is significant. And that's the first step you see on the one. If like everything is perfect and you go beyond expectation of what you were planning to do, you stop for efficacy. So it means it's weird, like you stop, so it's not good, but actually it's really good because you have an effect. If no, then you continue. And again, there you can stop on the full population or you can stop on the subpopulation because you're testing multiple things. If it's not the case, you continue, but then you test for futility on the other side. If your effect estimate is not larger than the boundaries, which is unlikely then to be significant at the end or having the promising effect, you should stop because you're exposing patients unnecessary to non-efficacious treatment. And so, again, it can be on the full population, on the subpopulation. And you've passed these two boundaries, then you continue, and then it can be S only, F only, and so on and so forth. And then you accumulate your second stage of patients, and then you combine the evidence from stage 1 and stage 2 to actually make the claim whether you're efficacious in the full population or in the subpopulation. So I couldn't resist to put you some stats there, but I won't go in detail today. So it's a one-sided test. So you look at the frequency of pathological response. You compare from your treatment to your placebo. So it's not 0.05, it's 0.205 to start with. But you have 2 stages, so you're making the test twice. So then it means you have to spend your alpha between stage 1 and stage 2, according to the sample size. So that's why you have an alpha 1, or 125, and then an alpha 2, or 1.84. So that's the first thing you do. But on top of it, you're multiple testing. So you have to adjust for that. So you can test for F, S, and F and S. So there's like procedures that are existing, closed test procedures that are very nice in the frequentist area, but you need to account for that. And then on top of it, as I was saying before, you need to have your futility boundaries to specify a priori. Okay. So that's how you plan it, but everything is pre-planned again. Like you cannot change when you do that. You cannot adapt your plan as you adapt your trial. So you have to perform by simulation, of course, as you can imagine, your power calculation to see whether that would pan out over a different scenario. The scenario there was whether PD-L1 was only prognostic, so acting both in the full PD-L1 positive or negative on whether Atezo was working on both, on the PD-L1 positive only, and so on and so forth with some differential things. Okay. And at the bottom, of course, all the analysis at stage 1 is done by an independent review board. The sponsors don't see that. They just tell you, okay, what proceeds to stage 2 and how. So just quickly, not to finish on that, simply the methodology. At the end of stage 2, so there was no stop for efficacy. Everything continued without futility. We had nice results on the ITT population, the intent to treat, so the full population. On the PD-L1 positive, that was significant, but that didn't pass the threshold of significance. No, that was like, okay, positive, but not significant. So the end of the conclusion was actually, atezolizumab was also acting on the PD-L1 negative population, so there was no reason not to have the label in the full population. I'm not picking that on by purpose, it's just what the data are telling you. Okay, so some learning about adaptive trial design there. In the confirmatory setting, because Paola was exemplifying that in the exploratory one or early clinical trial. So you can see that what we're trying to convey is that the stat methodology with all the things we have on simulation, like everything's in there to have the type 1 control error. So like no one should be afraid of that, that's okay. As long as you pre-plan accordingly, there's no problem in that. The real thing is more the second one to me is not so much about whether you have the right conclusion, is the estimation of the effect. And the estimation of the effect, of course, is key when you want to see the clinical benefit compared to the risk of any molecule. And that can lead to a lot of simulation that can go there. The reason, there are many factors that are playing on that. The first one is like, when do you do your stages? And if you do your first stage too early, you accumulate a small amount of data compared to the ones you want to have at the end. And if you stop for whatever reason for efficacy, the estimates you're going to have on this treatment effect is largely going to be biased because you have some sample variability and it's too good to be true. So you have to correct for that, and that's not easy. And of course, there are like multiple biases that can go into the play while you're accumulating data on the trial. The main thing, again, in the center, like, it's all about planning and pre-specification. It's not because it's adaptive that you can adapt as you go. No, everything has to be pre-planned, the boundaries, the decision rule, the number of stages, and so on and so forth. And then, of course, the trial and conduct integrity. What I want to say there is that, the information gathering at different stages really has to be limited to the IDMC, the Data Monitoring Coming Board, because if you start to have information leakage about this group doesn't work or this group, that can really play on against your integrity at the end and the conclusion you want to draw. But okay, I think the message there is that nothing is impossible, that can be done, it's done, and for the many reasons I was outlining. So, but in the last thing I want to say, like, precision medicine thinking, tools and things is much more than adaptive trial design. I'm saying that because that's most of my work. And the example I want to show is, again, the patient centricity, so you remember that. So the example I want to show you quickly is about safety monitoring. The context is the following. So we have a drug currently in development called glofitamab. It's a T-cell engager, so it's a bispecific T-cell. So on one side binding to T-cell, on the other side to CD20. It's developed in NHL, works really nicely, but I can't say too much about that. That's not the point anyway. The problem is that it's creating, at first infusion, cytokine release syndrome in about 20% of the patients. And it's dose dependent, as you can see on the right, and I will explain the graph a bit later. So it's not a good thing at all. It's not a good thing at all because, like, you have to manage that. That can be life-threatening if the grade is severe. And on top of it, for the patient, it means he has to stay overnight in the hospital because you cannot run the risk of having a cytokine release syndrome and sending the patient home and having a cytokine release syndrome in the next 24 hours. So you see on the complex graph on the right, the y-axis is actually the timing, and the x-axis are the different cohorts. These are data from phase 1, so you see different doses there, different scheduling, like step up and so on. But basically what you can see is that, in a nutshell, you're increasing the dose, you're reducing the time to event, really after first infusion, and as well as increasing the fraction and to some degree the severity. Okay, how can we do a better job? And it's all about, can we tackle the diversity? Can we look into that? Can we identify subgroup of patients that are at risk and the ones that are not? And if we can succeed on that, then we can say, okay, a fraction of patients can go home, the other one is staying in the hospital, and it's much better for everyone. So the data you're going to see are on phase 1. There are much more data, but I don't have the luxury of sharing non-published data, as was done before. So we're going to stick to that, and I will let you infer the rest of the story. So it's very easy when you do that. You have, first of all, something we always have to remind. You can only depend on the data you have. And the second aspect as well is that you always have to think that the multivariate score or the model you're going to build is something that you plan to have in the clinic. So it has to be simple and based on measurements that are actually at your disposal at the time of the patient taking the first infusion, because you can see you have 24, 48 hours and not much more. So that's what we did. I'm not going to go to all the factors, but you can see Gazyva. DZ is Gazyva, so it's a B-cell depleting agent that you can pre-treat your patient with. Extranodal involvement of the tumor, whether it's good to the nodes. You can have TNF-alpha at baseline, even though, that was surprising that was significant to me, but okay. LDH, white bone cold cells, B-cell lympholysis, age, and then some patient characteristic, cardiac morbidity, bone marrow infiltration of the tumor, peripheral infiltration stage of the disease, which is the disease severity, and so on and so forth. So don't go to the details there, and the other thing as well is that this is dose dependence. So the way we built that is a model that was thinking that the baseline factors were independent from the dose, but we have to adjust for the dose because we know it's the main source of variability. So we did that in a way in a surface response model, gathering some kind of statistical learning with variable selection and so on, and some bootstrap to look at the stability over that. In the training, we end up with like the variables that you see highlighted in blue. And then we make, as usual, a faith because a leap of faith. We didn't trust so much in the 196 patients, which is not a lot, in the weight we were estimating, so we didn't take the optimal weight, but we just scored them very simply 2, 1.5, depending on the size of the effect. And then we did a model validation that you see on the left, which is only to have the cutoff, so it's much smaller part. You don't change the variables, you don't change your score. You just try to see what is the cutoff that is giving you the nice property. In that case, you want to screen out. So you want to have maximizing the negative predicting value, which means if you're lower than the cut, you have very low chance to have the cytokine release syndrome, and you keep the fraction of patients that you isolate shouldn't be too small. And we converge on the famous cutoff of 5, which was giving you 50% of patients of low risk, and with the probability of having an event of 5%, which was deemed in discussion with clinicians and stakeholders to be manageable in the grand scheme of things. And you see the property of the positive prediction value on the 40%, so you have a much higher risk than the 20%. And again, on a specific dose, this data are abstracted for 2.5 milligrams. So we took the full dose range and we just say, okay, we only want to predict what would happen if. And then we had another cohort, completely independent, never seen the data, no data leakage of any strange things like that. Just look at the cutoff of 5, and to our nice surprise, again, the fraction of patients was the same. And then the NPV, or the 1 minus NPV, so the probability of not having the probability of having an event knowing that you're below the threshold was very low. The reason why I'm showing the data, of course, is that now we have many more cohorts, and that this result constantly is true. We try to reduce the score to make it even more simpler for clinicians. And we are also having a prospective trial piggybacking on existing cohorts that are recruiting to actually prospectively validate that, and then go to a regulatory agency at the end of the year. So the reason why I'm showing that is not to show a success, even though it's always pleasing. It's just to show that, again, precision medicine can really lead you to much more in your toolbox of innovation than adaptive trial design, like looking at variability and diversity, which is what agronomy is about, by the way, is really key to understand, decipher the complexity of human biology. That's very clear. The other statement is that it's not only about adaptive design and so on. There's a full toolbox. Paola, showing something, and Trishna will show you other example on that. And then cautious if you want to do this adaptive trial design. If you go in confirmatory, you need to really be clear on the questions you want to have, which is always true with clinical trials, and care in the number of stages and decisions, because only 2 stages, 2 groups can become complex. So imagine you multiply that by 5, then it can become really ugly in terms of decision rules and power calculation. And I thank you. I don't know how I did with time, but okay.
Thank you so much.